Ladies and gentlemen, you are all welcome to my YouTube channel. My name is Mukhtar Ali from the Department of Biochemistry, Ahmadu Bello University, Zaria. So now, in this video, we are going to look at the MCQ. The first question says, gluconeogenesis is also carried out in muscles and brain. A, true, B, false. So the answer to this question is actually false because muscles and brain lack an enzyme called glucose 6-phosphatase. It lacks an enzyme called glucose 6-phosphatase, and this is one of the major key enzymes in gluconeogenesis. So any organelle that doesn't have this enzyme, it will not undergo gluconeogenesis. And of course, the major points where gluconeogenesis takes place is kidney and liver, because kidney and liver have both glucose 6-phosphatase. So that is why gluconeogenesis takes place in both kidney and the liver. And then the next question is, name the enzymes which is responsible for the conversion of glucose 6 for the conversion of pyruvate to phosphoenol pyruvate. So this is one of the first steps of gluconeogenesis. First, remember that in glycolysis, an enzymes called pyruvate kinase converts pyruvate phosphoenol pyruvate, and the reaction is irreversible. So since it is irreversible, it means that phosphoenol pyruvate or no, okay, I forget. I just wanted to say the pyruvates, also phosphoenol pyruvate, is generated from phosphoenol pyruvate using an enzyme called pyruvate kinase, and that is the name of the enzymes. So the conversion of phosphoenol pyruvate to pyruvate is an irreversible step. So therefore, pyruvates cannot be converted back to phosphoenol pyruvate directly. So what happened in gluconeogenesis is that the pyruvates, before it converted to phosphoenol pyruvate, it follows some series of steps. Pyruvates will first be converted to oxaloacetate, and the oxaloacetate will be converted into phosphoenol pyruvates. And of course, gluconeogenesis, this is one of the reactions in gluconeogenesis, and gluconeogenesis for both in the cytoplasm, it occurs both in the cytoplasm and mitochondria. Because after glycolysis, the pyruvate generated is transported to the mitochondria where it converted into acetyl CoA4 TCA cycle to continue. So now if you have pyruvates in the mitochondria, there is no any transporter for So generally there is no any transporter for the oxaloacetate in the mitochondria. So now what happened? The pyruvate generated, which is present in the mitochondria, it will first be converted into oxaloacetate using an enzyme pyruvate carboxylase. Pyruvate carboxylase is an enzyme that adds CO2, that is carbon dioxide, to pyruvate. Pyruvate is a 3-carbon compound and oxaloacetate is a 4-carbon compound. So the enzymes pyruvate carboxylase add CO2 to pyruvate to generate oxaloacetate, and then this oxaloacetate that is generated in the mitochondria cannot be transferred or cannot be transported directly from the mitochondria to the cytosol because the mitochondrial membrane lack oxaloacetate transporter. So as a result of that, before this oxaloacetate is converted into phosphoenol pyruvate, it will first be converted into malate. So we have mallets, in the we have mallets enzymes. We have mallet enzymes, and this mallet enzyme is called mallet. The enzyme is called malate dehydrogenase. And this malate dehydrogenase is found both in the mitochondria and the cytosols. So the mitochondrial malate dehydrogenase will convert oxaloacetate to malate, and then the malate, because it has transporter on the membrane, then it will be transported to the cytoplasm. And in the cytoplasm, there is also another enzyme called malate dehydrogenase, the same enzymes in the cytosols. So the major functions of the cytosolic malate dehydrogenase is to convert this malate back to oxaloacetate. And then the oxaloacetate generated in the cytoplasm, then it will now be converted to phosphoenol pyruvate using an enzyme called pyruvate carboxykinase. So it means that carboxykinase is an enzyme that converts the pyruvate to phosphoenol pyruvate through oxaloacetate intermediate. So therefore, the answer to this question is pyruvate carboxylase. So now let's look at the next question. The next question says, which of the following is not a precursor of gluconeogenesis? So A, glycolytic product. Of course, the product of glycolysis is one of the substrate of gluconeogenesis. And examples of this product, we have pyruvate. Pyruvate is a product of gluconeogenesis, and it can be, it's a product of glycolytic fatty, and it can be converted to glucose through the process of gluconeogenesis. So it's a substrate of gluconeogenesis, and at the same time, it is the end product of glycolysis. And then, of course, B, citric acid intermediate. So remember, in the citric acids, you started with oxaloacetate. So oxaloacetate, remember, in the cycle, oxaloacetate in the cycle, the oxaloacetate in the cycle condense with acetyl-CoA. It condenses with acetyl-CoA to produce citrates. And the citrate will continue with the cycle. It will continue with the cycle until another oxaloacetate is generated. So it means that any intermediate between citrate to oxaloacetate, it means that it can actually be involved in the generation of oxaloacetate. And of course, on the previous questions, we saw how oxaloacetate is actually play a central role in gluconeogenesis. So therefore, citric acid cycle intermediate are precursor of gluconeogenesis. And of course, here, option C says gluconeogenic amino acids. So even from the word gluconeogenic or glucogenic, it means that there are amino acids that can synthesize glucose. So that is why from here we can simply understand that there are examples of gluconeogenic amino acid is an intermediate of gluconeogenesis or precursor or substrate of gluconeogenesis. An example of this gluconeogenic amino acid, we have alanine, glycine, are example of glucogenic amino acids. So then then the next question, which is question 4, says, what is the site of gluconeogenesis? That is, we have gluconeogenesis takes place. From our previous questions, we said that gluconeogenesis takes place both in the liver and the kidney. So therefore, here the answer is option E. So then the next question says, name the pathway for glucose synthesis by non-carbohydrate precursor. So the pathway for glucose synthesis by a non-carbohydrate precursor is gluconeogenesis. So when you synthesize glucose from non-carbohydrate precursor, so the process is called gluconeogenesis. And then 6, which of the following is a precursor of glycogen? Which of the following is a precursor of glycogen? So in glycogen, there are precursors that are used to add off or that are used to synthesize the glycogen, and of course the activated form of the glucose that is used in the glycogenesis is UDP glucose. So and then the next question says, the priming function in glycogen synthesis is carried out by A, lysine, B, arginine, C, glycogen and the glutamate. So generally, the glycogen in the liver is not usually, or is not exhausted after breaking down. It's not all the glucose that break down and utilized by the body. Some amount of the glucose will still remain as a form of glycogen, which can actually serve as a framing compound that will be used for the continuation of the synthesis of glycogen. And that glycogen that have the priming function is called glycogenin. It is called glycogenin. So option C here is the answer. The name given, the name will name the enzymes which is used for branching of glycogen. So of course, glycogen have both linear change and branching. So the linear change of glycogen is alpha 1,6 alpha 1,4 glycosidic one. That is the linear change, and the branch point of glycogen is alpha 1,6 glycosidic 1. So the enzymes that actually do the branching activity is called the branching enzymes. So branching enzymes here is the answer. And then, of course, the next question, now we are going to look at question 8. So which of the following hormone maintain blood glucose level by activation of gluconeogenesis? So generally, when blood glucose level is high, so you need to reduce the glucose, and that is done by the insulin. The insulin will actually activate the transport of glucose from the blood to the other peripheral tissues. And of course, whenever the blood glucose level is low, then the glucagon will be activated. And once the glucagon is activated, what next? What next if the glucagon is activated? It will now stimulate and activate gluconeogenesis to start synthesizing glucose so that it will be used to maintain the blood glucose level, because the blood glucose level is low. So gluconeogenesis is activated by glucagon. Then the chemical compound involved in the process of metabolism, so the compound or molecules that are used during metabolism is called metabolites. It is called metabolites. And then next, let's look at another question. So now the catabolic pathways is hexagonic in nature. A through B or false? The answer is true, because we have three types of metabolism. We have catabolism, anabolism, and amphibolism. So catabolism usually involves the breaking down of larger molecules into smaller ones with the release of energy. So energy is generally released during glycolysis, during catabolic pathways. So because energy is generated in the process of catabolism, the system or the pathway is called hexagonic. So the answer is true. But in the case of anabolism, it is endogonic, because during anabolic process, it's smaller molecules that join together to generate a larger one. So in the process where smaller molecules are assembling themselves, they utilize energy, they consume energy. So that's why they are called endogonic. So anabolism is endogonic, while catabolism is hexagonic. Then the next question says, which of the following has a spiral metabolic pathway? So generally, metabolic pathway have three nature. We have circular, like in the case of citric acid cycle, from the word citric acid cycle, glyoxylase cycle, they are all in cycle. And then we also have linear. It can be linear. So when we said it is linear, what linear means? It means that it is in a straight line. So glycolysis is an example of linear. And then we have spiral metabolic pathway, which is the fatty acid biosynthesis. So it usually spiral. So the answer is D. So now let's look at another question. So now the question here said the regulation of metabolic pathways is done by the following ways except one. So make the one. Okay, so now, sorry, mark the one. So now looking at this, we have substrate concentration. Of course, the concentration of a substrate of a particular pathway affect or can be used to regulate the pathway. Like, for example, in glycolysis, if you supply glucose, you expect a lot of glycolysis to take place. And if you deprive the cell from glucose, it means that there will be no glycolysis. So you can use the substrate to either upregulate metabolic pathways or downregulate metabolic pathways. And option B, allosteric regulation of an enzymes. Of course, enzymes, so many enzymes, apart from the active side, they have what you call allosteric side. So in the allosteric side, a substrate or a molecules can be bind. Often the binding of the molecules in the allosteric sites that can lead to either activation or inhibition of the enzymes. So therefore, can either increase or decrease the activity or the metabolic pathways activities based on allosteric activities. And then C, availability of oxygen. So you cannot use oxygen. Availability of oxygen do not actually use in the metabolic regulation. And then D, there is a extracellular signal. So hormones, growth factors like glucagon, epinephrine, and so many glucocorticoid, mineralocorticotropic hormones, all are examples of the chemical or extracellular signals that can actually influence or that can regulate the metabolic pathways. So here, the answer is C. The inhibition where end product of biosynthetic pathway inhibit the activity of the first enzyme. So generally in a pathway, we can have a multiple substrate like A here will be converted to B, and then B will be converted to C, and then the C will be converted to D, then E. So the E is the final product. So in each of these steps, there is enzyme. So this is enzyme 1, enzymes 2, enzymes 3, enzymes 4. So when the end product of the metabolic pathways inhibits the activity of the first enzymes in the pathway, it is called feedback inhibition. It's called feedback inhibition. Okay, so then name the type of the pathways which is involved in the synthesis of compound. So the pathways in which smaller molecules are used to generate a larger molecules, that is the synthetic pathways, is called anabolism. So therefore, anabolism is a synthetic pathways, and this is a degradative pathway, and this is amphibolism is degradative and the synthetic pathway. That is metabolism. So which of the following cycle shows? So the answer to this is option A. And then now, which of the following cycle shows metabolic pathway? So which pathway is an example of metabolic where both catabolism and anabolism take place simultaneously? So the answer is B. The answer is B. So then the next thing is to say thank you. Thank you for watching my video. And if this is the first time you are coming to my YouTube channel, please subscribe. Thank you.